Hey there! Getting ready for your Class 10 biology exams? Well, you are in exactly the right place. Today, we're going to break down one of the most important topics you'll face: how our cells divide and what it all means for our health.
Have you ever really thought about this? I mean, how does a single cell become, well, us? And how does life keep going generation after generation? The answer to both of these massive questions comes down to two amazing processes: mitosis, which is all about growth and repair, and meiosis, which is how we reproduce. These two are basically the engines of life itself.
So here's our game plan for today. We'll start with the life of a single cell. Then we'll jump into the two types of division. We're going to compare them side by side. And trust me, that's a huge exam topic. Then we'll touch on reproductive health and wrap it all up with a quick guide to help you absolutely nail your test. Let's get into it.
Okay, first things first. Before a cell can even think about dividing, it has to live its life. It has to grow and get everything ready. And this whole process, from start to finish, is what we call the cell cycle.
So what is the cell cycle, really? Just think of it as the complete biography of a cell, from the moment it's born until it's ready to divide into two new cells. It's broken down into two main parts: a really long preparation stage called interphase, and then a much shorter division stage called the M phase.
Now, what's pretty wild is the timing here. The actual division part, the M phase, is super quick. We're talking less than an hour. A cell spends almost its entire life in interphase just getting ready for that big moment. All the important stuff, all the prep work, happens right there.
An interphase isn't just one long, boring waiting game. Nope. It's a very active, carefully controlled process with three distinct steps: G1, S, and G2. You can think of it like a pilot's pre-flight checklist for the cell before it takes off.
So here's what's happening in each step. In G1, the cell just gets bigger, bulks up. Then comes the S phase, or synthesis phase. This is the absolute most important part. It's where the cell copies its entire DNA blueprint. Once that's done, there's no going back. The cell is committed to dividing. And finally, in G2, it doubles all its internal machinery and does one last check before division. All right, our cell has done its prep work and is ready to enter the M phase. The first type of division we're looking at is mitosis. Its job is really straightforward, but so important. It makes perfect, identical copies of cells. This is how we grow and how our bodies repair themselves, like when a cut on your skin heals. Mitosis happens in four very clear stages. And for your exams, you absolutely have to know this order by heart: prophase, metaphase, anaphase, and telophase. Let's quickly walk through what happens in each one.
Everything kicks off with prophase. Inside the cell's nucleus, all that long, stringy DNA starts to coil up super tight, forming those thick, X-shaped chromosomes that we can actually see with a microscope. At the same time, the wall around the nucleus, the nuclear membrane, starts to disappear, letting the chromosomes out.
Next up is metaphase. Here's a great way to remember this one: M for metaphase, M for middle. It's that simple. All the chromosomes get pulled into a perfect, single-file line right across the middle of the cell, what we call the equator. Each one is hooked up to these things called spindle fibers. And then anaphase. Here's another tip: A for anaphase, A for apart. Those fibers suddenly pull tight, and each X-shaped chromosome splits right down the middle into two identical halves. These halves then get yanked to opposite ends or poles of the cell.
Finally, we have telophase. A new nuclear membrane forms around each group of chromosomes at the poles. And then, in a final step called cytokinesis, the rest of the cell, the cytoplasm, literally pinches in the middle and splits in two. And the result? Two brand new daughter cells, each one a perfect clone of the original.
Okay, let's switch gears completely. We're moving on to our second type of cell division: meiosis. Now, if mitosis was all about making clones, meiosis is all about creating genetic diversity for reproduction. The key phrase to remember here is reduction division. Its whole purpose is to cut the number of chromosomes in half.
So here are the key things that make meiosis different. First, it only happens in our reproductive organs to make sperm or egg cells, which we call gametes. Second, it's not one division, it's two full rounds of division. And its main goal is to start with one cell that has two sets of chromosomes, we call that diploid, and end up with four unique cells that each have only one set of chromosomes, and we call that haploid.
Okay, listen up, because this is probably the most important slide for your exams.
They love to ask you to compare mitosis and meiosis. So understanding these differences is absolutely key to getting those top marks.
This table right here is your ultimate cheat sheet. Let's run through it. Mitosis, its purpose is growth and repair. It has one division, it makes two identical diploid cells, and the chromosome number stays the same. Meiosis, its purpose is reproduction. It has two divisions, it makes four unique haploid cells, and this is the big one, it halves the chromosome number.
All right, let's stop and think for a second. This is a classic exam question that really tests if you get the concept. What would happen if our gametes, our sperm and eggs, weren't haploid? What would the consequences be for the next generation?
And here's the answer. It all comes down to simple math. A healthy human being has 46 chromosomes. Meiosis makes sure the egg brings 23 chromosomes to the party, and the sperm brings the other 23. They combine and you get a new baby with the correct number, 46. If that number wasn't cut in half first, it would double with every single generation, and life just wouldn't work that way.
You know, understanding the biology of how life is created also means we have a responsibility to understand our reproductive health. So let's just briefly cover a few important points about making healthy and informed choices.
The source material points out a few key things we should all be aware of. First, there are legal ages for marriage, which are there to help ensure people are physically and mentally ready. It's also so important to know about sexually transmitted infections, or STIs. Some are bacterial, like gonorrhea, and others are viral, like HIV. Understanding how they're transmitted, and more importantly, how to prevent them with healthy practices, is a critical part of staying safe and healthy.
All right, let's bring it all home. We've gone through the science, now let's talk strategy. Here's your final checklist to make sure you are 100% ready for any question they throw at you on this topic. Okay, let's lock this in. 1. Know your definitions cold. 2. Practice drawing and labeling the stages of mitosis. They love diagram questions. 3. Master that sequence, PMAT. 4. That comparison table is pure gold, so memorize it. And number 5, be ready to explain the why. Don't just list facts, be ready to explain why meiosis is so essential for life as we know it.
So we've walked through the mechanics of how cells divide, but the really exciting stuff, the frontier of science, is figuring out the complex signals that tell a cell when to divide, when to stop, and what happens when those controls go wrong. That's a question for another day. Thank you so much for joining me, and best of luck on your exams.